Today I'd like to talk to you about my experience with learning through authentic practice. My class used something called Wisconsin Fast Plants in order to help us learn more about inheritance and genetics. Here's a quick video to let you know what fast plants are. Oh, now wait a minute. We didn't actually start with the fast plants. We started, well, I think we need to back up a little. We started out talking about tigers. These tigers, to be specific. Our instructor put the pictures of these tigers up onto a PowerPoint and asked the students to break into groups and identify characteristics and variances that they see among the tigers in their physical appearance. Once we identified different types of variations, we were asked to figure out how the tigers got to be the way that they are. Why do they look this way? And how can we explain the variation in the species? We explain the differences between the tigers by saying, well, you get different things from your parents. But then we found out that these tigers were in fact siblings. Then we looked at their mom and dad. Mom was a normal colored tiger, while dad was a white tiger. That's two variances, but among the cubs we've got three. We've got one normal looking tiger, and two of the tawny or golden tigers, and one white tiger. Although we had some general idea of the genetics that were involved here to get these types of variances, nobody could really pin it down. So that's when we took a step back and we talked about Gregor Mendel, the father of genetics. We did a web quest about Gregor Mendel. We learned a lot about his biography and background, including the fact that he couldn't pass the national teaching exam. So that's why he joined the monastery. We looked pretty closely at Mendel's research. We looked at what he observed when he crossed plants. We tried to hypothesize about what he would get with each generation. And we also tried to hypothesize about what was going on. We learned about Mendel's golden ratio of about 3 to 1, meaning that there's a 3 in 4 chance of producing one variance and then a 1 in 4 chance of producing the other, depending on which one's dominant or recessive. After studying Mendel and his work, we applied our knowledge to our own Wisconsin fast plants. Now I can show you a video about what the plants actually were.
Hi, I'm Angela White, a biologist, product developer at Carolina Biological Supply Company, and a former high school biology teacher. Today I'll be introducing Wisconsin fast plants. Fast plants are only available from Carolina and have been developed in partnership with the University of Wisconsin. These plants are perfect for busy teachers because they have a fast generation cycle of 35 to 40 days. From one day to the next, your students will see the rapid growth of these amazing plants. Let's begin with the fact that fast plants are indeed fast. If your students plant seeds on Monday, by Wednesday they'll have small green seedlings. In just over 2 weeks, the plants will begin flowering, and your students can pollinate them. In another 3 weeks, seed pods will begin to mature, and students can harvest seeds. Students can study the life cycle of a flowering plant from seed to seed in one grading period.
The plants are really neat, and while we were planting them, we were trying to determine what possible outcomes could result as the plants grew and predict the probability of each variance. The Wisconsin fast plants are very similar to the peas in that many of them only have 2 possible variances for a characteristic. As our plants grew, we started to look at multiple allele traits and also common dominant and recessive traits found in human beings, things like a widow's peak and a cleft chin. Then we also moved into mutations, learning about sickle cell and cystic fibrosis. Some of our plants did in fact show mutation at the end of their growth cycle. So in the end, I had the opportunity to participate in a very worthwhile and educative scientific learning activity. Our teacher actually employed many of the thoughts and tools that are recommended by our textbook. We use charts and graphs to track the growth of our plants throughout their life cycle. We also use Punnett squares in order to predict the probability of certain variances appearing in the phenotypes of our plants. We also used a lot of visual aids as we were learning about mutations, such as the sickle cell. It's a lot easier to look at a picture of a sickle cell and understand what can happen rather than to just be told. And some of the data or information that was tied to visual aids was just invaluable to have a picture there because it was so much easier and so much less confusing to have that visual aid available than it would have been to just try to make sense of it all if it had all been written out. I remember this lesson so well because of the pedagogy that was involved. We were given the opportunity to engage in authentic practice. Students were also allowed to work cooperatively. There were some other aspects of this lesson that really benefited me greatly as a student. Our teacher incorporated technology by providing us with a web quest that included a Web 2.0 application that would allow us to manipulate the genetics of fictitious plants to see how things would work out. This helped us get a deeper understanding of the content. Finally, our teacher incorporated a great deal of inquiry-based learning. She would ask us questions and allow us to work together in groups to come up with possible solutions. By allowing us students to do the intellectual work rather than just providing us with answers, we gained a deeper understanding and were able to formulate our own understanding as it related to our own knowledge. So I would highly encourage any teacher to incorporate these ideas in their own instruction in the future.